Curation Coalition database and some updates, but I'll also start with a little bit of an overview because it's been about a year since I talked about the GenCC, and I know that ClinGen has wonderful groups of curators who join all of the time. So just to get everyone on the same page. And I just wanted to point out that our preprint on the GenCC is actually available and medRxiv, and Jenny had linked that very nicely in the email talking about this presentation. So today what I'll be talking about is a refresher about GenCC and why we formed and the members of the GenCC, and talking about the Delphi survey that we performed to harmonize clinical validity terms. Then I'll talk about the GenCC database, which was launched in December 2020, and the updates that we've had since the last time I talked. I'll talk a little bit about the conflict analysis that we've done within the GenCC to resolve some of the conflicts now that we have the database to compare our assertions, and then also future plans for enhancing the database. I'm going to move this bar so I can see. Okay. So we all know that there's a great need for curated gene databases for gene disease validity. And this is actually really highlighted well by this graph here on the left, where I've plotted the number of genes for 14 different hypertrophic cardiomyopathy tests from different US clinical labs from the genetic testing registry. And you can see that the number of genes for each test varies widely, from maybe the lowest one having less than 20, maybe 18 genes, to some having over 100, maybe 100 to 110 to 115 genes. So this really points out that there are standards and consensus needed for which genes are valid disease genes that are ready for clinical testing. I know in the past, it had really been more genes is a better deal, and a lot of labs had potentially put on as many genes as they possibly could into their tests to make it seem like their test was a better deal. But then you get these variants back in these genes that have questionable gene disease evidence, and what do you do with them when you're potentially returning them to patients with clinical phenotypes? Something we also need to think about is there are potentially different levels of evidence that are needed for different clinical uses, such as predictive tests or diagnostic panels, exome genome analysis, maybe population screening in individuals that aren't affected with conditions yet. You would want to be sure that the level of evidence for gene disease relationships is maybe a little bit higher than those individuals getting diagnostic panels because they've come to attention because of clinical phenotypes, for example. And I should mention that in 2020, I believe, ACMG did put out a publication with some suggestions for ClinGen clinical validity levels for different types of testing. Bean et al., feel free to take a look at that. But their suggestion was that for diagnostic panels where people are clinically affected, you should have moderate and above genes on those. And for predictive tests or screening types of tests, you should probably consider strong and definitive genes only. And of course, if you're looking at research or anything in that realm, you can look at all different genes. But this really did point out that we needed some standards and consensus about gene disease relationships and databases. So we formed the GenCC, or the Gene Curation Coalition, and we've steadily grown in membership over the years. I think it was formed in 2018 at this point. And so we have public gene level resources, and those are all listed here. Not all of these submit to the database. Of course, like HTNC doesn't necessarily submit to our database because they don't perform curations, but everyone, and GKB just recently joined too. But everyone does weigh in on a lot of our decisions for setting standards, for example. So a lot of these are the key stakeholders, even if they don't necessarily perform curation, to be helping us set standards in gene disease validity. And then also importantly, we have private gene level resources. So many clinical testing labs that perform lots and lots of high volume clinical testing, therefore they've had to perform gene disease relationship curation, and they have a very beautiful database or extensive databases of gene curation that hasn't been shared anywhere until we had this database. And so many of these are partners with us too, as stakeholders, to help guide some of these decisions and really bump up our numbers in our database. So we had a few goals when we started the GenCC. First, we wanted to start by understanding the approaches and classification systems of the different curation efforts. We couldn't really compare if it was apples to oranges, and so we really need to put ourselves on the same page. And figure out who the audiences were for each, how the curations were done, et cetera. After we got to know each other a little bit, we wanted to work on developing consistent terminology. And so the development that I'll talk about today is for clinical validity buckets, so validity assessment. And then we also worked on consistent terminology for inheritance, allelic requirement, and mechanism of disease. And I'll talk about that a little bit in the enhancements at the very end of the presentation. Then we really wanted to post gene curations from any group willing to share with the public. So our database, which we launched in December 2020, is like a ClinVar for genes. It's a database of assertions for gene disease validity relationships. And ClinGen is one of the first submitters to the GenCC database. So once we had this database up and running, we wanted to be able to use it to resolve differences and collaborate on gene curation projects, which we're starting to do now. So first, when we were getting to know each other, we wanted to do some comparisons across groups. And some groups were a little bit easier to compare than others. For example, Panelap has 2 instances. One is based in the UK in Genomics England, and one is based in Australia for Australian Genomics. And so because they're based on the same platform, they have very similar curation styles. And so what they wanted to do was compare and see how concordant they were to just give us a sense. And so this is just an example of their comparison that Australian Genomics had 1,800 or so genes, and Genomics England had 2,700 or so genes. And so they compared the overlapping curations that they had, because they are 2 separate instances, and reviewed over 1,000 gene discrepancies and made some changes. So for Panelap, they answer the question, which genes should be on which panels for which phenotype. So they added 800 new genes to their panels based on this comparison. And they upgraded or downgraded 250 existing genes based on the comparisons that they were able to do together, really just showing that this comparison is very important, and sharing data for genes, much like sharing data for variants, is very critical so that everyone can be on the same page. And this was published, I believe, in mid to late 2021. We also did a gene curation bake-off where some of the original members listed up here of the GenCC performed curations on some of these genes listed here, and then we compared all of our curations. So you can see they're kind of color-coded. That green is the ones in the green rectangles are really the ones that are the higher level of evidence, whereas the ones with yellow and orange are kind of mid-level, maybe limited moderates in ClinGen speak, and then the red ones are like disputed, refuted, really shouldn't be a gene disease relationship. And you can see for many genes, we had a lot of discrepancies. There was a bit of confusion, and there's also a lot of term discrepancies. So this bake-off really showed us that we needed to start working together to sharing our information and also sharing our definitions of what we meant when we were saying these gene curation clinical validity levels to try to harmonize everything. And these comparisons did identify some reasons why we were discordant. Sometimes our definition of disease was different. Maybe some groups were really only, like ClinGen, for example, were really only considering high penetrance versus inclusion of lower penetrance phenotypes. I know that's been a difficulty. Potentially the purpose of curation was a little different, like ClinGen does gene disease validity versus Panelap's Australia and Genomics England that I just talked about include based on a phenotype for a particular gene panel. So that's a pretty different style of curation there, which may cause discordance. And then also differences in disease or phenotype assignment. As you know, with lumping and splitting, genes often have claims for multiple diseases. So maybe we weren't even evaluating the same disease. And that's some of the difficulty that we're having now when we're looking at our discrepancies in the database. And I'll talk about that a little bit too. And then I think one of the easiest ones to resolve by sharing data is the date of evidence evaluation. As you know, things are published rapidly, things change over time. So some curations were just older than other curations, and that could be the discordance. So understanding these differences and helping to resolve them really did require harmonization of terms and definitions first. And these were the comparisons of terms when we first started the GenCC. And you can see it's very confusing. For example, 2 different efforts actually had opposite colors for meaning different things. So in the one effort, red meant strong or definitive, and the other effort, red meant limited, disputed, refuted. whereas other people used words, other people used yes or no. It was just very confusing. So we really thought that we needed to do something to harmonize these terms because we couldn't put this all on a website. It would be very confusing for others. So what we did is we performed a Delphi survey, and a Delphi survey is an iterative survey to help choose clinical validity terms. And so we did a couple different phases to perform this Delphi survey. For the first part, we developed the survey. So we all worked together to come up with harmonized definitions. So we were all on the same page. We all meant the exact same thing for the strengths of evidence that did not contain the proposed clinical validity terms because we didn't want to lead anyone in a particular direction. In the second phase, we prioritized. So in a Delphi survey format, you suggest as many terms as possible, and then in further phases, you narrow down the terms by scoring the terms or rejecting the terms or accepting the terms. So first, we solicited terms from all the GenCC members, and then we performed our first survey. The GenCC members, 33, expressed their level of agreement, disagreement, or neutrality for solicited terms. We used a modified Likert scale, and they were also prompted to add additional terms for the second round of survey. So for phase 3, we refined, and we used iterative surveys to refine the term list. For the second survey, we extended the survey to both GenCC members and their curation staff. So for ClinGen, potentially even staff biocurators, and this was 38 people. So after the survey, we actually assigned scores to all of the Likert scale. So for the positive values of the Likert scale, we used positive numbers, and for the negative values of the Likert scale, like disagree, strongly disagree, for example, we used negative numbers. And for each term, we summed those numbers. And then we made a standard deviation across all of the terms and eliminated those terms with scores 2 standard deviations below the mean, suggesting that they had highly negative opinions of those scores. And that was how we came up with a final amount of terms to survey the international genetics community in survey 3, where we surveyed 241 individuals across a range of different communities internationally to determine the final term list. And we also recorded an optional 10-minute explanatory video to pretty much give you the overview that I just gave you. And so for phase 4, it was consensus and implementation. So surveys 2 and 3, so the GenCC members and their curation staff and the international genetics community had 93% term agreement. So that really suggested that the choices of the GenCC, the final choices, were in agreement with the larger genetics community. So we use these terms to standardize submissions to the GenCC database with the addition of the category supportive for less granular submissions. So the 2 submitters to the database, one of them is not live yet, that use supportive are those that you would expect, OMIM and Orphanet, because really there's just presence or absence in that database suggesting gene disease relationships. And these are just some demographics from survey 3 when we surveyed the international genetics community. So locations of all the respondents here. These were the populations that we surveyed. So European Society of Human Genetics, American Society of Human Genetics, Canadian College of Medical Genetics, ClinGen, British societies, molecular pathology. We also put this up on Twitter, Australian Genomics, Panel App, for example. And then we also asked people if they participated in consortiums, and multiple responses were allowed. And we were actually happy to see that most people did not participate in a consortium because we didn't want them necessarily to be biased or too close to any of the development of gene disease validity curation or terms that they would be biased in their answers. So most people were not part of consortium, which was helpful. We also asked them about their familiarity with genetics in case we had to stratify answers based on that. And most people were very familiar or relatively familiar with genetics, so we didn't have to worry about that. And when we asked people to describe themselves with multiple responses allowed, most people were researchers or clinical genetics laboratory directors or staff. medical genetics physicians, and so on. So these are the finalized terms here, and ClinGen has adopted these terms entirely, which is why they are exactly the same. Definitive, strong, moderate, limited. Animal model only is a subset, at least for us, of no known disease relationship. Disputed evidence and refuted evidence. And this is basically what I just said previously, that all of this is used for submission to the GenCC website. So now I'm going to talk about updates to the GenCC database. And I really wanted to thank Scott Geisinger, who has moved on from ClinGen, but did a lot of the seminal work for the GenCC database, really designing it, and Phil, who's on the call, who has taken the helm and is now helping me with updates and keeping the database up to date. So these are the statistics, and if you saw the last presentation for the Gene Curation Coalition database, you'll see that it's grown a lot. At this point, we have over 15,000 submitted classifications on about 4,600 or so genes from 12 submitters. And the database itself is search.thegencc.org, and these are just the summaries of all of the classifications that we have. As I mentioned, supportive is only used for 2 submitters. As of right now, these 5,300 or so submissions are from Orphanet. OMIM is not in the database yet because they are hoping to have an API so that their data set is not forked or out of date. But these are all the clinical validity buckets, and you'll notice these are all the harmonized terms that we came up with from the Delphi survey. Just to give you a little bit of use and summary statistics. So this is a map of users by country with the top 7 countries listed as of September 2021. So we have pretty good use distribution, with the US being top user, China, UK, Germany, Australia, Israel, and Canada being the top 7 countries. And then to give you a sense, between January 1st and September 10th, there were 36,470 page views by 4,126 users. I think at this point we have over 250 registrants to our email list that we send updates to, such as large updates in the size of the database, updates on features, and then hopefully when our publication is published, we'll also send that update to them. To give you some more summary statistics, we thought this was interesting. This is a graph of the number of submissions per gene. And just to remind you, we have about 4,600 genes. The average here, as you can see, based on the graph, is about 2 submissions. And interestingly, the max is 35 submissions. I can't remember off the top of my head which gene this was, but I can tell you that 33 is FGFR2 or 3, if I remember correctly, which has 33 assertions on that one entry. So to give you some screenshots and walk you through the database now, this is the gene list landing page. So when you go to the search.thegencc.org, you'll come here, and it will just show an unfiltered gene list, and you have lots of abilities to filter it. So you'll see all of the terms along the top, all the clinical validity buckets. All of these are checkable or uncheckable. So if you only wanted to see definitive ones, for example, you would uncheck all of the other ones. You can filter by gene symbol. As of now, it does have to be the most current HGNC gene symbol. We're hoping to work with aliases in the future, but just keep that in mind. You can also search by a particular disease. I'll talk about in the enhancements in the future, a little bit more flexibility with disease searching. As of right now, the keyword that you type in does have to actually be present somewhere in the disease name for this to work. So keep that in mind. And then you can also filter by submitter in the dropdown for all the submitters. If you click on, there are a couple different help buttons. If you click on About GenCC Classifications, it will tell you about the Delphi survey that I just told you about, and then also give you all of the standardized definitions that we use for our Delphi survey. So each line is a gene. It will tell you the number of submitters for that particular gene, and then we'll also show you the tally of the types of submissions that are available for a particular gene. If you were to click this little arrow, it will open up the gene just to a slightly more informative expanded view here. So it will show you the submitters. It will show you what they've classified for and what disease. So here, what we're saying is disease equivalence, and you'll see this a little bit more on the gene level pages I show you. We accept for our submissions orphanet, OMIM, and monarch disease ontology or MONDO terms for disease identifiers. However, in order to kind of group them the best that we can, we actually use MONDO to group together diseases. So for disease equivalence, we are meaning those MONDO terms that are used to group together submissions. So you can see here, there's one disease equivalent. It's very easy. They're all submitted under Noonan syndrome. But for disease equivalence, that is the MONDO term that we use to group together multiple submissions, even if they have submitted with an OMIM term or an orphanet term. We use the mapped to MONDO term for everything to kind of harmonize together. So if you click the details button, if you want to know more about a particular submission than just this little quick dropdown, it will take you to a gene-specific page. And the tabs here will show you there are a couple different ways that you can sort things. So the first one that we're on is by classification. We're hoping to add a little bit more in this gene facts page up at the top, but as of now, it just has the gene symbol, really, and the return to list button. For each tab, you can filter by different things. You can filter by classifications if you only wanted to see definitive, for example. You can filter by diseases. It will make things disappear. This could be particularly useful for FGFR2 or FGFR3 that had 33 assertions to be able to filter here. You can filter by mode of inheritance and submitters. So for each line, you'll see we have the color theme going throughout, but you'll see the clinical validity bucket, you'll see the gene name, you'll see the disease here. And we'll talk about this a little bit more on the disease page. But for example, ClinGen submitted using the MONDO term here, but you can see that Invitae submitted using the OMIM term. And so we do provide that transparency that it was mapped to this MONDO term here, but it was submitted as this OMIM term. You'll see the mode of inheritance with a hover over for the abbreviations and what they mean. And you'll see when it was evaluated at the top, which is most important, and then in gray when it was submitted. You'll see who submitted it. If there's a public report, there's a link out. So ClinGen links to the ClinGen website and the particular entry. You can click to see their assertion criteria, much like in ClinVar, that in our case would be the latest gene curation SOP. And if you want to see more information about that particular entry, you can click to the more detailed submission level button, which we'll get to in a couple pages. So when you go to the next tab, it sorts it by disease. This is a different gene, but to be able to see, it groups by each particular disease. And I chose this one actually because I wanted to show you a little the difficulty that we have that even though some assertions aren't on the very exact same disease term, you can clearly tell that we're making an assertion on the same disease. It's just the terms that people choose to use. So here's the example here. Arterial calcification of infancy, which was orphanet, and they submitted as their ORPHA term, which was mapped to just the general arterial calcification of infancy, whereas TGMI G2P used arterial calcification generalized of infancy 2. And this was submitted using the OMIM term, which is associated particularly with this gene. So you could see that you would want to do the comparison on these two, but if you were to do it entirely automated, you wouldn't necessarily see that these assertions were the same. So this is a little bit of the difficulty that we're coming across now with gene disease assertions and disease naming and which ones are the same and which ones are different. So we're taking a couple different approaches for that, and I'll talk about that. But it also involves some manual review and curation too. And these top lines here are all MONDO disease equivalents, as I mentioned, but we do talk about which term they used for originally submitting. And you, of course, can link out to that term using this little click button. Then you can also sort by submitter, and each of these pages gives you the exact same pieces of information, just different ways to look at them. So if you click out to read more, for example, or more details in any of these, you'll get to the individual submission page. And this can be really helpful I think I mentioned this in a slide or two for ClinGen curators, if you're curating a gene-disease relationship, because sometimes there'll be evidence or notes. So Illumina has some really nice evidence summaries like ClinGen does. And many groups at least have PubMed IDs. So you can look at the PubMed IDs that were used to assert this gene-disease relationship. If you want to learn a little bit more information on the submitter page, we've mimicked ClinVar for our submitter pages. So this is just the ClinGen example here. And it will tell you a little bit about that effort. It will tell you their website. And then also importantly, if you ever had any questions about submissions for that particular submitter, we have a contact information for the coordinator for that submitter. So I'm the contact for ClinGen submissions. And then we also have a link for assertion criteria if you wanted to learn how they perform their gene-disease relationships. And then a nice summary of all the classifications visualized for that particular submitter. So this is relatively close to all of ClinGen's classifications. We do perform submissions to the database every so often. So we do suggest that people still search on the ClinGen website instead of GenCC, because until we have an API, if someone publishes something to the ClinGen website, we might have just done a submission to the GenCC database, so it might be slightly out of sync. There's also the ability to download data. So there are a couple of different formats to download the data. When OMIM is available, you'll be able to download everyone's data but OMIM's data, because OMIM does require a license for data download. So just to point that out. And then a new feature that I wanted to point out is GenCC curations are also now displayed on the ClinGen website. So if you search a gene, for example, I searched GJB3 here. If you click the button View Gene Facts, it will open up. I highly encourage people to go to the Gene Facts section. I didn't even really know it was there for the longest time, but it has some really useful information here, like nomad scores, very helpful. Main select transcript, also very helpful. A little blurb about the function, particulars, aliases or previous symbols, just all very helpful. But what I wanted to point out was the GenCC classifications here. So it will show you all of the GenCC classifications for that particular gene. And then if you wanted to go read more about them, you just click this and it will take you to that gene specific link for GenCC. And I should point out that this is available for genes that we haven't curated to. So it doesn't necessarily have to have this ClinGen curation data attached to it. It could just be a gene that you're interested in, as long as it's a gene, and you can search that and find these gene facts and find if GenCC has classified it. So particularly helpful for ClinGen curators who are curating gene-disease relationships. So what I suggest, if you have a gene-disease relationship that you are assigned in a ClinGen GSEP, is to search the ClinGen website for your gene of interest. We pull in data for genes that haven't yet been curated. You can use that gene facts section. It's really helpful. It will give you the main transcript. It will give you nomad scores, and it will tell you if GenCC has curated the gene at all. So either way, if you go straight to the GenCC database or you link from ClinGen, you can see, as long as the disease appears correct, you want to determine if it's a completely different inheritance pattern or disease from one that you were assigned. You can click on the individual submissions to use the PubMed IDs from those curations. I usually don't use the assertions directly, but you would want to use the primary evidence they cite. And I should mention that some groups do use stronger definitive assertions in the GenCC database to justify scoring functional evidence. One of the main examples being protein interaction, and saying that an interacting partner was classified as strong or definitive in the GenCC database, so that you didn't have to necessarily curate both genes simultaneously. But please do double check with your GSEP on that front. So now that I gave you an overview of the GenCC database and what we've been up to, I wanted to talk about how we've started to work on resolving conflicts. So as I pointed out in previous slides, we are having some difficulty because even though people may mean the exact same gene, they may not submit on the exact same, or sorry, when I say gene disease, they may not submit on the exact same disease term. So it's very difficult for 15,000 gene curations to programmatically calculate conflicts if the diseases aren't the same. So we've come up with a tiered approach for this, starting with the most granular and moving to the least granular. So the first conflict type is pretty simple. We call these exact match conflicts. So these are ones that the gene, the disease, and the mode of inheritance all match. So the disease term matches exactly, mondo term for mondo term. And this is an example here where it's the same gene for two submitters, same disease, literally, same mode of inheritance. And what we're counting as conflicts here are anything limited and below versus anything moderate and above as a conflict. Kind of per that Bean et al. Paper that I talked about, that moderate genes and above should go on clinical testing panels and limited and below should not. So if everything matches and one submitter says limited and one submitter says strong, this is counted as a conflict. And we're calculating this at the gene level instead of the number of conflicts per gene, because we just have to look at the gene as a whole anyway and try to resolve it within the GenCC. So out of 4,600 or so genes, 246 of them have this exact match conflict status. So it's 5.4% of the database. To move to a less granular way to define conflicts, we're calling the next one gene conflicts. So the gene matches, but we're not looking at the mode of inheritance or the disease term. And this is an example. So the gene matches for the two submitters. The disease is completely different. We're ignoring it. The mode of inheritance is completely different. We're ignoring it. But we're just looking at one person said moderate and one person said limited. And this is the least granular approach. You would imagine that most of the time, as per this example, this would identify an incorrect conflict, but at least these entries are being flagged for us so that I can go in and manually look at them and see if there are any true conflicts within these entries. And so if this is the most granular approach, 5.4% conflicting, this is the least granular approach, we were relieved to see that really only maximally 13% of the database is conflicting. So this is 610 genes here that may have some sort of conflict, but likely many of them, much like this example, are not true conflicts. So I think that the real percentage of conflict lies somewhere in between these two. And somewhere in between is the gene and mode of inheritance conflicts. So in these cases, we made sure that the gene and the mode of inheritance matched. We ignored the disease to try to get to the root of the OMIM term with a number after it versus just the general disease term, like the example that I showed you. So this example, the gene is the same. The disease might be the same, but the terms are not exactly the same. So with inherited retinal dystrophy and microcornea, myopic chorioretinal atrophy, telecanthus syndrome, not exactly the same clearly, but one might just be higher in the mondo tree. Mode of inheritance is the same. And then one person is saying strong and one person is saying limited. So it's technically a conflict. So as you would expect, this falls in between the really specific conflicts and the really unspecific conflicts. So this is 488 genes here, 10.7% of the database. So I think this is probably a little bit closer to the real percent of conflicts within the database. But we were happy to see that the database itself overall isn't highly conflicting with our submissions. So to work through resolving these conflicts, I've worked with Larry Babb to generate some conflict reports. So we started off with the easiest set, the exact match conflicts, because I didn't have to really spend much time looking at the disease terms and more just the overall attributes of each submission. So each submitter did receive a report with exact match conflicts last month or so. And these are the conflicts here that I mentioned, limited and below versus moderate and above. Supportive is actually not counted within this because it's just not granular enough. So the reasons that a particular submission was counted as a conflict in these kind of simpler conflict reports, we took the outlier approach, which ClinGen often uses with invariant curation. Stephen Harrison has done a lot of this work, that if a particular submitter is an outlier, it is most likely that they should reevaluate theirs first because it might be incorrect. So for example, if 2 submitters assert strong and 1 asserts limited, the limited one is considered the outlier. And often in these cases, the outlier is older, or at least submitted at the same time. If the outlier is newer, that's a bit of a flag. And so I'll often flag all 3 submitters in this case and just tell them to email amongst themselves, maybe some evidence is missing, or to all reevaluate, maybe you missed a piece of literature. If there are only 2 conflicts, I usually flag the older one, thinking that that person missed literature or missed pieces of evidence. And then more complex conflicts, which I'll show you an example, either have multiple submitters flagged or we have to talk about them on a call. So this is an example. Each month now we start to talk about more complex examples of these conflicts on the GenCC calls to help try to decide and resolve them. So this is an example here, CAM K2G, and you'll notice that things are kind of all over the place here. So if in this case we were looking at exact match disease terms, you'll see that 3 individuals classified this as limited and 2 individuals actually classified this as strong or definitive. So that's pretty significant of a conflict and no easy way to tell who was correct because they're kind of all over the place as far as dates. So we had to present this one on a call. So for conflict discussion, how we usually do it is I start by presenting an overview of the gene and all the combined evidence for the curation. And then I give each submitter a notice ahead of time so they're not surprised that their curation is on a call and they can prep potentially information or a little bit of an explanation ahead of time of how they came to their conclusion, and they can comment on the curation on the call. And then usually the combined members of the GenCC decide on an appropriate evidence level based on all the combined evidence in this discussion. And usually because we don't want to tell people what to do, they have to go back and reevaluate using their assertion criteria, et cetera. We started to go with a less granular decision like moderate and above or limited and below per our conflict calculations to offer guidance to the submitters. And then submitters use this guidance and evidence to reevaluate their curations. And we can't force someone to agree with us or agree with our decisions. So we have to respect their final classification when they resubmit, because the way that they curate might be different, or they might be asking, should something be on a panel or not? So this is kind of the way that we guide these conflict discussions for more complex conflicts. So now I'm going to spend a few minutes on future database enhancements, and then I'll open it up to questions. So the first one that we'd like to have as an enhancement is an API. As I mentioned, OMIM does not want to submit to the database currently because their entries change daily, nightly, very, very often. So pretty much as soon as they would do a spreadsheet submission, and that's what we have in place right now, it would be out of date. Or would they really be able to submit all of their assertions within a particular spreadsheet? It would just be inconvenient. So we'll be putting a submitter API in place to ingest OMIM data. We're actually going to use the ClinGen data as the first test case for this. And then we're hoping to have a user API in place later in 2022. So as you know, we just have downloads right now of spreadsheets, different formats. So we're hoping that people will be able to plug into our API at a certain point for clinical pipelines, whatever they need later in 2022. But we'd like to have the submitter API in place first before the user API is in place. We're also hoping, much like the ClinGen website, to have a login screen in the dashboard. So in this case, you would be able to save searches, you'd be able to follow genes, you'd be able to filter and choose how you would like to potentially receive your data within the API. Say you just wanted definitive genes, for example, or just genes that were asserted after a certain date. You could save all of this information within your login screen and dashboard. And then another thing that we think is very important and will greatly improve the searchability of the website is disease searching flexibility. So we're hoping to incorporate, because our database hinges on Mondo, even though it accepts submissions from Orphanet, OMIM, and Mondo as far as disease terms, we'd like to incorporate the Mondo hierarchies into the database. So this will really allow users search flexibility for disease terms. So you won't need to make sure that you actually have a term that's included within a particular disease name to actually pull it up. You should be able to search by any term in the Mondo tree, synonyms, maybe potentially even in the future HPO terms. And this is also being discussed for the ClinGen website. So we're hoping potentially to pilot on one site versus the other and then implement on both sites. And then hopefully sometime soon in 2022, we're hoping to granularly capture mechanism of disease. We know that some curation groups are actually already capturing misinformation in their curation. So as we've come up with standardized ways to describe mechanism of disease, so the terms for mechanism of disease, we'll be able to granularly capture this. And a lot of this work has been done by James Ware and Angarad Roberts and their team at TGMI. And then an interesting thing that actually Phil doesn't even really know about yet, but we have to talk about, we are hoping to have a special submitter that we take care of that actually points out candidate genes. And what I mean by candidate genes are those genes that are in ClinVar that have likely path or path variants, but they're not in the GenCC database and they're not even in OMIM yet. So there's not a ton of these genes actually, but we'd like to include this type of submitter within the database in case people are looking for candidate genes that they should keep an eye on to make our database a little bit more complete. And potentially either ClinGen could prioritize these within the general GSEP or particular GSEPs as up and coming genes potentially, or even other submitters can look at these and potentially curate those gene disease relationships. And with that, I will end there. And I wanted to thank the steering committee for GenCC, all of the members for really submitting so much data that's so useful to the community. And then Scott and Phil for the website and database, because we wouldn't have such a beautiful website without them. And that's all I have. So I can stop there and take any questions or comments.
That was fantastic, Marina. Thank you so much. What a huge amount of work has gone into this, and just so helpful to have all of this information together. Does anyone have any questions for Marina? I think your presentation was so clear and so nice. I really liked your suggestion though about using the GenCC website to look for classifications for genes for our ClinGen curations for example, protein interaction. I think that's really nice because yes, as you mentioned, if you're going to use the protein interaction data, you have to be able to say that that other gene product, the gene for that has been assigned I think a definitive relationship with the disease. So this is really nice to be able to refer to the GenCC website for that, if your GSEP is okay with it, of course. But yeah, I really like that suggestion.
Yeah, I think it's important that a lot of expert panels just check in with that. I know the syndromic disorders GSEP already does that because our gene list is so wide ranging and kind of all over the place with its genes that we, and Illumina is a submitter of the GenCC too, that we often do just use the GenCC database for the stronger definitive assertions for that.
Yeah, it's really nice. It's a great idea. I was really interested to hear what you were saying too about the candidate genes, Marina. I guess that hadn't really crossed my mind before, that there might be genes with path or likely path classifications in ClinVar that aren't yet even on OMIM. So yeah, that's really neat to sort of think about those and prioritizing for curation as well.
Yeah, I'm trying to remember. So we had some discussions with an international group that I want to say their group created something like Gene Alert or Genome Alert, and they actually brought that to our attention, that there were a number of genes that weren't in OMIM but did have path or GenCC and did have path or likely path assertions. I don't think it was an extremely high number, but it was a little bit higher than I thought. Like I think it's like 50 to 75 genes.
Wow. Wow. And presumably all sort of reported recently, or
I would assume so.
Yeah. Great. So does anyone have any questions or thoughts on the GenCC website. Oh, I see a question in the chat. Thanks for this presentation. Do you have any plans to include other submitters or databases, for example, SysID for intellectual disability?
That's a great question. And that was actually one of the few reviews that we did get for our paper that recently came back, is to kind of clarify how we accept submitters to the GenCC database. And we do have a little bit of information on our frequently asked questions in the database. But generally, if a group contacts us and asks, can they submit to the database, we have a couple things that we ask them just to make sure that their data are granular enough to be included in the database. So usually, A, they have to be willing to publicly share their data, which if it's a publicly available database is fine. B, they have to have a rigorous gene curation method that's usually on par with Strand et al. So the ClinGen method. And C, they have to be able to map all of their gene curations to our harmonized clinical validity bucket terms. We really have only left supportive for OMIM and ORFNet because they are pretty much seminal in the field and original members, but everyone else has to be able to map to definitive, strong, moderate, et cetera. So those are kind of the requirements that you would have to submit or even to become a member of the GenCC in general and start to weigh in our discussions, and then also submit to the database.
And here's another question. Are all the approved ClinGen gene disease associations automatically added to GenCC?
The best that we can. So as of right now, our submission is spreadsheet based. So we only submit the ClinGen data, the entirety of the data set every so often. So there is a slightly out of sync difference between the ClinGen website and the GenCC website because there's no API at the moment, but that will definitely be our first API test case. So that basically once you publish a gene curation to the ClinGen website, it should be in the GenCC relatively quickly. As of now, there's a little bit of a lag.
Okay, so any other